thank you again for the introduction. I'm with the Office of Science and Technology, Health and Ecological Criteria Division, Human Health Risk Assessment Branch. So the goal of my branch is basically do risk assessments related to microbes and chemicals. Thank you, Hilary, for inviting me to speak today about the human health effects from exposure to cyanotoxins in drinking and recreational waters. I gave this presentation back at the beginning of this year, and it got very good feedback. So I hope you like it as well. Thank you. Shane, please, next. So I will start this presentation with summarizing the adverse health effects from exposure to 3 common cyanotoxins in the US. And then I will provide a very briefly overview of the EPA's approach to deal with harmful algal blooms in freshwater systems. Next. So if you were able to hear from the presenters yesterday, and you also heard from Ingrid Karus yesterday, you have a great overview of what are harmful algal blooms, and more specifically about cyanobacteria and freshwater systems. The focus of my presentation today is on how we see the problem of harmful algal blooms from the One Health perspective, meaning how HABs affects people, animals, and the environment, which could lead to economic consequences. Today, I'm going to focus on the effect on humans, right? And how an interdisciplinary approach with toxicologists, microbiologists, ecologists, epidemiologists, veterinarians, engineers, and many others is needed to prevent and manage these impacts and achieve optimal health outcomes. Next slide, please. Regarding the adverse impacts on human health, illnesses from exposure to cyanotoxins vary depending on the toxin type and the dose you are exposed to. People can be exposed to cyanotoxins several ways. You can be exposed through the skin to cyanobacteria cells and their toxins while you're recreating in toxin contaminated water. You can breathe toxins in aerosols or tiny airborne droplets that are wind-blown in spray. You can ingest cyanobacteria and their toxins incidentally while recreating, or you can ingest cyanotoxins in drinking water or contaminated food. Next, please. Another example of a route of exposure is through intravenous exposure. I forgot to include it in the slide. For example, via dialysis with contaminated water. It is a rare exposure to cyanotoxins, but unfortunately, a case of contamination event of a dialysis clinic, actually 2 cases of dialysis clinics several years ago in Brazil, resulted in several deaths in that rare event. So, and even though it's rare, it could happen, and it's another exposure type. We currently do not have adequate health-based data for some routes of exposure to cyanotoxins to determine what is the health risk to that exposure type. The majority of the available data is on the ingestion exposure to cyanotoxins, which is the one that I will discuss today. So in 2015, in collaboration with Health Canada, we developed risk assessment for 3 cyanotoxins: Microcystins, Anatoxin-a, and Cylindrospermopsin. Why these 3 toxins? These 3 toxins at that time were included in the candidate contaminant list, or CCL, which is the program that EPA follows for determining what is the risk for non-regulated contaminants. So we included these 3 cyanotoxins, we evaluate their health effects, we determine their occurrence in drinking water systems, and we make what we call a regulatory determination. We decide that we need a regulation, we decide we don't need a regulation, or we don't have the data to make a regulatory determination at that moment. So we needed the health effects or risk assessments for these 3 particular toxins. This risk assessment comprised a summary of acute, short-term, and long-term health effects, as well as carcinogenic effects from exposure to these toxins, as they are reported in epidemiological studies, toxicity studies, and case reports. And we use that data to determine what is that dose response that will cause those specific adverse effects. And we use that to derive a reference dose that will be used in the development of guideline values. In drinking water, we develop health advisories, and for recreational exposure, we develop recreational criteria and swimming advisories. So that was a very compact overview of our risk assessment process and regulatory process and the development of guideline values. In the following slides, I will discuss the findings on the adverse effects from ingestion to these cyanotoxins. So next slide, please. So we will start with microcystins. We know that the majority of the information that was published, majority of the toxicological data is on microcystin LR. Therefore, the evaluation of health effects from exposure to microcystin that we did is based on studies that evaluated that and only a specific congener. When microcystin is ingested, it's transported throughout the body in the circulatory system. Once it reaches the liver, microcystins strongly bind to cytosolic proteins called protein phosphatases 1 and 2, or also known as PP1 and PP2, which result in the retention in the liver. So you ingest microcystin and they find the liver, and that's their paradise. They stay there. They inhibit that enzyme activity of the PP1 and PP2, and that results in liver toxicity. The limited data in humans are mostly from the accidental ingestion of toxin-contaminated water during recreational activities. These reports, or human reports, what we call case reports, demonstrate that exposure to microcystins could affect the liver. So we determined that microcystin is a hepatotoxin. However, as with many human reports, we don't have data that could be used by risk assessors to develop that dose response and a reference dose, because you usually do not have cyanotoxins concentrations, and there's always that possibility of co-exposure of the people affected with other microorganisms. So we use laboratory studies. The laboratory studies of microcystin LR have found adverse effects on the liver and also have found effects on the reproductive system. Liver effects are the health effects more common and are shown as an increase in liver weight, bleeding in the liver, and an increase in liver enzymes in the blood. The reproductive effects were reported by a single study in 2011, showing effects in ovaries and testes, decreasing sperm counts, and a reduction in sperm motility. We evaluated that study. We sent it for peer review. We got comments from our peer reviewers that supported that the study itself had a lot of limitations in the design of the study, in the reporting, and in the methods of the analysis that were used. After the development of the risk assessment in 2015, new studies on the toxicity of microcystin LR on the reproductive system were published. We evaluated that data and we found the same adverse effects in both female and male systems, but none of the studies were able to characterize the mechanisms of these toxic effects. So how microcystin is able to affect both reproductive systems. So therefore, we understand that this is a gray area that needs to be further research for microcystin. There are two chronic studies published on the long-term effects from oral exposure to microcystins, and both are limited in the design and data reported. One study was done over 18 months and used only one dose, therefore we couldn't find a dose response. And in that dose, they didn't find any toxicity. And the second study was done for 280 days, but did not report quantitative data on the effects observed. Therefore, the adverse effects from long-term exposure to microcystin in drinking water is unknown. Next slide. We also evaluated the carcinogenic potential of these three cyanotoxins. So for microcystin, there are several human epidemiological studies that have reported an association between microcystin in drinking water and the occurrence of liver and colon cancer. These studies have some limitations. For example, some were ecological studies, and with ecological studies, you're not able to prove cause-effects associations. Some had poor measures of exposure, potential co-exposure to microbial or chemical contaminants, and in some cases, a failure to control for known liver and colorectal risk factors, like, for example, smoking. Animal studies suggest that microcystin is a tumor promoter. Microcystin LR is classified as group 2B by the International Agency for Research on Cancer, which is possible carcinogenic to humans. But the limitations in the database, for example, not having an adequate long-term dose response studies, doesn't allow the EPA to determine the carcinogenicity of microcystins. Next slide. The next toxin is cylindrospermopsin. Although the mechanism of the toxicity of cylindrospermopsin has not been fully characterized, we know that once ingested, cylindrospermopsin is again transported throughout the body in the circulatory system, and it gets to the liver, and like microcystin, attached to the liver. However, cylindrospermopsin binds to a different enzyme type of enzyme system, which is the cytochrome P450, and inhibit the hepatic protein synthesis. So it's affecting both the liver and the kidney, different than microcystin that only affects the liver. Human data on the toxicity of cylindrospermopsin is from an outbreak in Australia caused by short-term consumption of drinking water. The major adverse effect observed was kidney damage. Data on the concentrations of cylindrospermopsin were not reported, and there were high concentrations of copper sulfate in the water. Therefore, there was a disconfounding factor that could have influenced the observed symptoms. Acute and subchronic animal studies have found kidney effects, such as an increase in kidney weight, decrease in protein creatinine ratio due to glomerular dysfunction, and also liver effects. These liver effects were observed, but only at the highest doses, which makes the kidneys the most sensitive organ to cylindrospermopsin damage. In our review, we couldn't find any chronic studies of oral exposure to cylindrospermopsin. Therefore, we don't know what are the adverse effects from exposure or long-term exposure to cylindrospermopsin. Next slide. Several laboratory studies have studied the potential damage to DNA by cylindrospermopsin. However, up to date and following EPA's guidelines for carcinogenicity risk assessment, there are no bioassays or long-term study evaluating the cancer potential of cylindrospermopsin with dose response data, or studies that are longer than 2 years with more than one species with multiple doses and evaluating different endpoints. Therefore, the current data is inadequate to determine the carcinogenicity of cylindrospermopsin. Next slide. The last toxin I will be discussing today is Anatoxin-a. Anatoxin-a has been associated to the death of livestock and dogs exposed to contaminated water. However, linking the exposure to the effect is usually difficult to document, as Anatoxin-a undergoes rapid degradation in the presence of sunlight, which makes it hard to detect during animal poisoning events. We know that once Anatoxin-a is ingested and distributed in the blood, Anatoxin-a attached to neuromuscular nicotinic receptors located in the brain, and it starts a psychological response, a physiological response in the central nervous system. So different than microcystin and cylindrospermopsin, Anatoxin-a is a neurotoxin, so affects your nervous system. It will not attach to the liver, it will attach to the central nervous system. Once it binds to those receptors, Anatoxin-a stimulates the muscle and cause fatigue, muscular twitching, and paralysis. That continued stimulus follows a respiratory arrest, which will lead to a rapid death. There are no human data, case reports of oral toxicity of anatoxins, thank God. There's one single dose response study found deaths occur within 2 minutes of administration of anatoxin, which tell us that anatoxin has a high acute oral toxicity. The animals show loss of coordination, muscular twitching, and death by respiratory paralysis. There are 2 short-term subchronic studies. The studies are limited because evaluated a few endpoints, and endpoints that are not related to neurotoxicity, which again, as I said, when you have a hepatotoxin, you evaluate serum chemistry, you evaluate liver enzymes in your blood. With a neurotoxicity, that's not the endpoint that you're following. You're following the endpoints of locomotor, twitching, etc., or effects that are represented in your nervous system. These studies also provided limited quantitative data and used partially purified extract. One study found several minor hematology and blood chemistry changes that were not considered to be toxicological significant and reported a death of a single mouse that couldn't be explained. In the second study, the highest dose did not cause any adverse effects. Therefore, when we did this risk assessment, we determined that the available information for Anatoxin-A is inadequate for us to determine what is the reference dose for a reference or guidance value. No studies have been done to determine the chronic toxicity from exposure to Anatoxin-A. Next slide. And there's no tox information on carcinogenicity. Next slide. Another exposure we evaluated was the accidental oral ingestion of cyanobacteria cells during recreational activities. Epidemiological studies have found associations between exposure to cyanobacteria and human health effects, including dermal and allergenic reactions. However, when we evaluated these epidemiological studies, we observed that the relationship between the cell density exposure and specific health outcomes like dermal symptoms, eye, ear infection, irritation, fever, GI illnesses, respiratory symptoms were not consistent. So we could have a study that found ear irritation when it was exposed to 5,000 cyanobacterial cells per milliliter, and then we find the same ear irritation when it was exposed to 100,000 cyanobacterial cells per milliliter. Some of them didn't have information on the cyanobacterial community composition. So therefore we understand that to develop a reference dose based on cyanobacterial cells is not adequate at this moment until new studies come that have some consistency on the results. Next slide, please. Okay, so I'm in the summary. As a summary, another click, please. The adverse effects from exposure to microcystins from oral exposures are liver toxicity, and for cylindrospermopsin are liver and kidney toxicity. Next. The main toxic effect of Anatoxin-A is acute neurotoxicity. Based on the limited toxicity data, EPA was not able to derive a reference dose for Anatoxin-A and to develop a health advisory. Next, exposure to cyanobacteria cells, yes, is consistent with an increase in adverse inflammatory effects, such as skin, eye, or ear irritation, fever, GI illnesses, and respiratory symptoms. And at this time, as I mentioned, EPA understands that available data is insufficient to develop quantitative recreational values for total cyanobacterial cell density. Next.
3-minute warning.
Thank you. We identified several research needs. They're listed here. I will highlight that we don't know what are the risks from exposure to mixture of cyanotoxins, and we don't know what are the risks from inhalation or dermal exposure, or have a dose response that we could use to develop guideline values for those specific exposure pathways. Next slide. Very briefly, I included here other adverse effects from exposure to cyanotoxins. I always get these questions. They have been associated with the death of wildlife and domestic animals. We understand that ingestion of water compared to their body weight, the pets usually shows rapidly symptoms and maybe immediately, especially if exposed to Anatoxin-A, have a response immediate. Fish could be exposed to cyanotoxins through the food web, or directly feed on cestone, or through dissolved toxins in the water. From the review of the literature, we know that different fish species have different uptake routes and metabolisms. We also learned that cyanotoxins can bioaccumulate in organs and tissues, but tend to bioaccumulate more or higher in the liver and hepatopancreas. And lastly, irrigation water contaminated with cyanobacterial blooms can contaminate irrigated crop and plants. It could get all the way to the grains, fruits, and leaves. It's usually more concentrations in the roots, but the literature on this topic is very, very rare. Next slide. It's very limited. So as I mentioned in the beginning, at EPA, we address the HABs issue following a One Health approach. Our actions include multidisciplinary professionals that develop guidance and recommendations, conduct assessments and monitoring. We do outreach and promote communication, creating partnerships and collaborations, and doing research. Next slide. So next. As I mentioned, we develop guidelines and recommendations. Currently, there are no federal regulations for cyanotoxins. We developed 2 health advisories for drinking water for microcystin and cylindrospermopsin. We develop a recommended recreational criteria and swimming advisories for microcystin and cylindrospermopsin. Along with that, we develop a series of recommendation documents and supplemental technical guidance documents to help the states with the application, if they choose to, of health advisories and recommended criteria. Next, please. We also do assessments with the unregulated contaminant monitoring rule. We have conducted a monitoring of 10 cyanotoxins in drinking water systems from March 2018 to September 2020. As I mentioned, we use that data and health data to make regulatory determinations. We also do National Aquatic Resource Surveys, or NARS, and I'm pretty sure that this group is very aware of those type of surveys that have included cyanotoxins in the National Lakes Assessment and other assessments like the Coastal Assessment. Next slide. We do outreach and communications. We're going to be talking about communications a little bit later, but EPA keeps the Cyanobacterial HABs website. We publish a monthly newsletter. We conduct national webinars like the one we did last week and the following week for the tribes. And for the past 5 years, we have held regional workshops across the United States. Next slide. We are very involved in partnerships with several and federal and non-federal work groups, and our regional offices, a great team of great professionals, also collaborate with the states, agencies, and universities. And lastly, next, we do research, which is mostly done by the Office of Research and Development. My last slide is my contact information. Oh, here, I wanted to show you this beautiful dog. Her name is Pandora. Pandora is one of 6 dogs that were exposed to a Microcystis bloom in the summer of 2018 in Martin County, Florida. Pandora was really sick, and although she was really sick, thanks to the prompt veterinary care, Pandora and other 4 dogs survived that event, and since then she has been an actively advocate for clean water in Florida. That's it for me. Thanks for your attention.